Hi, this is Miss Linton, and this is my wonderful APBio class reviewing Chapter 36, Body Fluids. But before we do that, this is what I want to do. Before we do that, I want to talk a little PCR with you. I've got orange on here. Ish. Okay. On your PCR, if you're having trouble like doing the fragments, stranger danger. If you're having trouble doing the fragments, you know, it's the DNA, it's that page, that handout I gave you. You remember that handout? Yes. Okay. And you have the first one that's on here, whatever it is, it's like a block, right? And this has something like 38,800 something. 200? I think so. Okay. So, and then I put on the first reverse primer for you. It's super annoying because it's reverse primer, so you have to read it backwards. I put it on there for you so you can see how to do that. Okay, and then maybe there's one that's listed here. I don't know what it is, 38,000 something. Okay, what you have to do to figure out how long that one section is you have to go back and see where does it start right here. So if this is 38,200, this is 38,199, 98, 97, 96. You see what I'm saying? Figure out what this number is right here where it starts. Figure out what that number is. You with me? And then you figure out what this number is right here. So whatever number it gives you right here, figure out how many nitrogen space, figure out what this number is. Right? This number will be bigger, right? Let's call this A, and let's call this number right here B. And if you take A minus B, then you'll know how long this fragment is. Do you follow me on that? You'll know how long that total fragment is. Does that make sense? Okay. And then you do the same thing with the other two primers. So I gave you the first one, and then all you have to do is figure out what numbers, the two end numbers. And where people make a mistake is they don't include that, but that gets photocopied too. So you got to go from one end of the primer to the other end of the primer, and then use the numbers there to figure out what the two numbers are. And that's why your first one is like 201 or 202 or something like that. Then once you know this number, write this number down. Whatever this number is, write it down, and then where your next one goes on there, then figure out what number that is at the very end and subtract that from it, and then you'll know how big that piece is. Right? And then you can line it up with a marker in the photograph, yes? Are you okay with that? You need more time, or are you going to be okay? I'm asking you. What? More time, or are you going to be okay? You want more time to talk to me tomorrow? You want me to collect it on, what's next time? Thursday? Would you like me to do that? Yeah. Okay. Well, let's see. Thursday, I'm not even here. Why don't I make it the end of day Thursday? Because I'm not here on Thursday. And then you can like have time to visit about it. Does that sound a good idea? Okay. End of the day on Thursday. Will that make your evening better? Okay. Who loves you? That's right. Okay. So remind me, what did you say?
I said in a non-teacher way.
Oh, okay. Remind me before you leave to change that on Google Classroom. Okay? Could you please right now tweet, text all your friends? I can make a what? Yeah, I will. Right, you can help them. Okay. I guess I can go like this. PCR. Why isn't it writing? Now, due Thursday, 3 p.m. All right. Very good. And we're recording all that. That's really good. Let me pause for a second. All right, here we go. Now, the first part, I had you watch a video on this, right? Yes? You watched the video? Yeah. Okay. So there's parts of it you need to know, and parts are just in there just kind of to use as examples. So the big part here is about, when you talk about excretion, we're really talking about body fluid regulation. And if you remember what amino acid structure is, the first step when you're going to deal with an amino acid is you could burn it through the process of cellular respiration. But before you can burn it, keep calling me APBio. Okay, before you can burn it, you have to do what? What's the process called? You know what it is when you remove that group? Deamination. You remember that? Okay, deamination is when you remove the amino group. Now, what you do with that amino group is going to depend on what kind of organism you have, how you develop, if it's live birth or you're going to develop in an egg, and how much water you have. Now, of the three, of ammonia, urea, and uric acid, ammonia is the most toxic. So if you're going to excrete ammonia, you have to have a lot of water available to you in order to excrete that. So if you're a saltwater organism, would that work? No, because if you're living in a saltwater area, probably the saltwater is hypertonic and you are hypotonic, you're going to be very concerned with keeping all of your water. Does that make sense? Okay, so you need a lot of water. So that's why if you look at this chart, you have to have a whole bunch of water if you're going to excrete ammonia, but this is very low energy because all you have to do is deaminate it and then you're done with it. You will have to invest some energy into it if you want to convert it to urea, which is what we do, but you don't need as much water. You can use less water, but you have to use energy because you're going to take a carbon dioxide and two ammonia groups and then form urea. Where in our body does urea formation take place? Yes, in our liver. Okay, so once we eat our food and digest our food, where does the absorption of those nutrients take place? Small intestines. And then we're going to leave the small intestines, the blood is going to leave the small intestines via the hepatic portal vein and go right on over to the liver. And there, that's where your amino acids can be deaminated. You can snip off the ammonia group and then hook it up with another ammonia group and a carbon dioxide, and lo and behold, you'll have urea. And then the liver can put urea in the blood, right there at the liver, go back to the heart, go to the lungs, go to the heart, back out to the body again, and if it happens to go to the kidney, then it will be filtered out due to glomerular filtration, and it'll stay in the P tube, right? The only chance of us reabsorbing a little of that urea back into our blood would be at what part of the nephron? The collecting duct, right? Because it's so concentrated by the time it gets to the collecting duct that some of it can kind of diffuse back into the blood. All right, now that's not going to work though if you're a bird. Because if you're a bird, using yolk, which is the nutrients, right? Because you know the yolk of the egg is the nutrients of it, and it's slowly consuming the nutrients from the yolk. And some of those nutrients are obviously going to be amino acids, and the baby chick's liver is going to have to do deamination of the nitrogenous group there, that ammonia group. He doesn't have fluid he can put in the egg. If he had fluid that he was excreting, if he was peeing in his egg, he would drown in his own urine, right? It would crack the egg open that he generated that urine. So he has to invest more energy, in fact the most energy, but this is the least amount of water needed if you form uric acid. And then you can store it inside the egg. If you're a bird, it's lightweight. You don't have to have this big heavy bladder of urine. You have very light uric acid, and you can just literally poop it out with all of your undigested or unabsorbed food. So that's why like bird poop, if you've ever seen one on a car, or maybe you're at the beach at your nephew's birthday party and maybe a seagull flies over and does his black tar crap right onto your head, if let's say that ever happened, and you go to the bathroom and they don't even have, because they're being light and they're not trying to make paper towels, so there's only the blow dryer. So your friend has to take toilet paper to try to pull the black tar crap out of your hair. That could happen. And So their poo is a mixture of their undigested, unabsorbed food as well as their uric acid. That's good times. Okay, and so you can see pictures of what kind of organisms and what they make. Okay, so we can see that here as well. All right. Then the next part is all mini-me. So this is just showing you planaria, who you're familiar with the flame cells. That's very typical planaria, keep the water moving. Earthworms are the closest to us. They have nephridia. They just have multiple all along. Each segment has 2 nephridia, which are very similar to our nephrons. And an earthworm, in order to keep their skin moist, they just pee out the side. It's like they're peeing. They have 2 bladders in each segment, and they just pee out the side. And that helps keep their respiratory surface, which is their skin, moist. What a benefit. Okay? And so you can see that here, and you can see blood pathway, pee pathway, just like us. Open circulatory system, insects use Malpighian tubules. Okay, so aquatic organisms, if you live in salt water, you tend to lose water and gain salts, but in fresh water, you tend to gain water and lose your own salts because your body is hypertonic to the fresh water around you. So salts are going to go from a higher concentration to a lower concentration. When you look at small invertebrates and sharks, the way they do that is that they try to make their bodies isotonic to the fluid in which they live, and sharks achieve that by keeping a high urea content in their blood. When you look at marine fish versus saltwater, this is freshwater fish, and this one is a marine fish right here. If you look to see what happens to them, if you live in salt water, what's salt going to tend to do? If you live in salt water, what is salt going to tend to do? You're living in a very salty environment. What will salt tend to do? Yeah, it'll tend to come in. And you'll get it from the water that you drink. So what you're going to want to do is actively excrete. If you're a saltwater fish, you're going to want to actively excrete it at your gills to get that salt out. And then you're going to lose some water because you know what's happening at the gills. What's happening at the gills? The countercurrent exchange. And there's water in your blood, and that water is going to leave your blood, and it's going to go right out into that salty. And you can see right here, you pee very little because you can't afford to give it a lot of water. The opposite of that happens in freshwater fish. They don't have to drink. Water just wants to go into them because they're hypertonic relative to their environment. So, and they lose salt, so they have to actively take up salt. So you can see why this is why you can't put a freshwater fish in a saltwater tank and a saltwater fish in a freshwater because it's going to be really bad. And they can have a lot of urine. The best fish regulators are salmon because they move between a freshwater and saltwater environment. But this is a nice little chart, kind of compares them as well. Okay, so that is the evolution of that. We talk about terrestrial organisms have a great tendency to lose water. Think of us as if we were a plant, and think about when we follow water in a plant, what do we call it? Water potential, right? What would you say about a plant? It would lose water, right? Why? Because the water potential inside the plant is higher than the water potential out in the dry air, right? Same thing for us, right? The water in our bodies is greater than the water content out in the air. So water is always going to want to go from a higher water potential to a lower water potential. We lose water all the time. Every time you exhale, you're losing some water, right? That's why you can clean your sunglasses, because you're exhaling some of the water. Why do you have water on your breath? Because in your alveoli of your lungs, you have to keep water in there to allow for that gas exchange, for that diffusion to take place. We're always afraid of losing water. And so organisms have different strategies on how they get water in their body. In the food that you eat, and metabolism makes water, right? Who catches at the bottom? Oxygen forming water. So that's where we can generate water besides what we eat. How do we lose water? It evaporates from our skin, it's in our feces, so our feces will move along, and then excreting, peeing. Those are all the ways we lose water. Things like kangaroo rats, they never even drink water their whole life. They live in a desert environment. They get all the water they need from the grains that they eat. Some things like seagulls that might poo on your head, they have a salt gland where they actively excrete salt out of their bodies. When you look at some skin adaptations to prevent water loss, they could have a thick, scaly skin. You're gonna learn about the water bear. I don't know when it's Thursday in your ed puzzle. You're gonna learn about the water bear and how they can just shut down and just let all the water out of themselves, and they just chill for years and years and years until they get a watering environment again. Camel nose, they have a countercurrent exchange in their nose to recover moisture. That's another place you have countercurrent. And this brings us to the human urinary system. So, in the human urinary system, we want to remember the structures and their function. So you have the kidney. What's the job of the kidney? Filtration of the urine. Okay? And they do let's name those steps of urine formation. Glomerular filtration, tubular reabsorption, tubular secretion. Okay, that's all going on, and then we're excreting whatever's left over. We filter the blood at the first set of capillaries. We take back what's important at the second set of capillaries. And then we actively transport waste that we don't want also into the second set of capillaries. Whatever waste we have is going to travel from the pelvis of our kidneys down our ureters. It's going to get stored in our bladder and then our urethra. Why do we have a bladder? It's similar to the reason why we have a stomach. Storage. So we're not constantly urinating on ourselves. Right? We urinate where we want to urinate. Okay? And when we want to urinate. And then the urethra is out. When you look at a cross-section of the kidney, outermost part is the cortex, then the medulla, and then the pelvis. And embedded in the cortex and the medulla, what is the functional unit of our kidney? The nephron. Yeah, the nephron is our functional unit that filters our blood. So we could follow blood flow, and we could follow urine flow, and we talked about at the first set of capillaries, it's like when a woman or somebody's cleaning out their purse, they dump everything out, good stuff and bad stuff. And then you, in the second set of capillaries, start taking things back, and then whatever you don't take back, then that's what you're going to pee out. So when we looked at the nephron, this is the most simple way of looking at the nephron, but we want to just review the blood flow. So let's do that. We're at the renal artery, and then it's going to break into the what? Say it. Afferent arterial. What's something the afferent arterial can do that has to do with maintaining homeostasis? What could it secrete? Starts with an R. Renin. Remember the renin-aldosterone connection? Okay. So then it's in afferent arterial, and then it goes into the glomerulus, which is a ball of capillaries that sits in the Bowman's capsule, and another name for it is the glomerular capsule. And so there, at that first set of capillaries, you dump everything out. Now, obviously, you can't dump out red blood cells or white blood cells. If you're dumping out red blood cells, your urine would be red, right? Okay? And if I went and punch, right into the, then if I did that a bunch, he would pee red. Why? Because I've broken a lot of his capillaries inside of his kidney, and so due to the hydrostatic pressure coming in there, or if he had high blood pressure, that could also cause him to damage his glomeruli, and then he would pee blood. But he doesn't. Okay, the blood vessel leaving that is the efferent arterial, and then the efferent arterial is going to travel into the peritubular capillaries surrounding the nephron, taking back what's important, putting into the urine what the blood doesn't want, and then it's going to leave via the renal vein. Okay, and if we analyze it specifically, we just want to make sure we know what's happening at each region. So we have glomerular, do this with me, glomerular filtration. So what's going out at this point? What kind of things are we pushing out? Tell me what the everything is. Is it red blood cells? No. Is it white blood cells? No. Is it water? Is it sugar? Yes. Okay, it could be nutrients, it could be salts, right. Okay, so everything goes out and it goes into the what? Glomerular capsule. Then that goes twisty-turny and it becomes the what? Proximal convoluted tubule. Say it. Proximal convoluted tubules. Now, at the proximal convoluted tubules, what is getting actively pulled out and put back into the blood? Yeah. No, but they're learning. What actively gets put back in there is you'll have some sugar that gets reabsorbed, right? But what else? What do we really have? Salt and water. So I always remember salt and water, right? Do this with me. Salt and water. And then when you go down the loop of Henle, which one? The descending, we're doing only water. Then up the ascending is only salt. And then again, salt and water. And then you put into the P anything that's bad. And then you go down the collecting duct and it's only what? Water. And a little bit maybe of urea. Okay, and that's the key thing that you need to remember. Okay, it's everything's out. These two are both salts and waters. Only water. Like the two ones that go down are the water ones, right? This one is just purely water. This one is water and a little bit of urea. How come water doesn't come out of the ascending loop? It's impermeable. And then it turns the corner, goes down the collecting duct. There's some hormones that can influence that collecting duct. Can you name something that could influence that collecting duct? Antidiuretic hormone? Exactly. Antidiuretic hormone makes the collecting duct more leaky because I don't want to die. If I have ADH, okay, if I have ADH, then I will reabsorb even more water. Okay, if I have ADH. Because I don't want to die, I need more water back in my blood. Hello. See the children learning?
Wonderful. This is such an awesome sight.
And then this is...
And they're not here out of fear or anything, are they?
No, we were recording you. Do you need me to sign something?
Yeah, I do, please. Do you want to have your autograph?
There we go. You are so welcome.
Thanks so much. I'll leave you to learn now.
Okay. So, and then the third thing that happens here is tubular, oh, thank you. Thank you, thank you. Tubular secretion, and this is where you're actively taking stuff like creatine or things like that or other wastes or drugs that you have, and you are actively transporting them into the blood. Are you okay with that? Are you stressed about it, or you got it? To me, it seems kind of easy once you, yeah? Okay. Because the thing I would ask on a quiz is, what happens at this place? What happens at this place? What happens at this place? So you want to make sure you know. Yes?
So the creatine and like drugs, that gets back into the blood, or is that in the urea?
You're taking it out of the blood and putting it into the urine. Yeah. Because you're like, I don't want this in my blood. And some of it came back to me, or it didn't get out, or it didn't get filtered out. I want to make sure it's out. I don't want it.
Answering secretion?
That's tubular secretion. Because if I'm secreting something, that's going to take energy. Filtration is due to hydrostatic pressure. The reabsorption we do is, you know you can't pump salt. I mean, you know you can't pump water, but you can pump what? Salt, and then you get water to follow. Good. All right? When we look at how it maintains homeostasis, these are our metabolic wastes right here. Primarily, it's going to be urea for us. And we know where we get rid of urea. Urea is going to happen at the glomerulus. Creatine, we can do at the distal convoluted tubules. Uric acid could come out too, but if we have too much, it could crystallize. You know, we don't want to do that. Water and salt balance, that's all happening in those paratubular capillaries. Are you getting the water back that you need? Now, what can influence that? We have some hormones that can influence that. Aldosterone can influence that. ADH from our pituitary can influence how much water we reabsorb as well. pH, if we want to, on the tubules, we can secrete whatever we need to keep our pH just right, because it's monitoring. It's taking the pH as that fluid is passing. You can say, oh, I need to push out more hydrogen ions, I need to actively transport those out, or I need some bicarbonate. So you can regulate that. The hormone part is the part that sometimes wait, let me do a couple other things. You okay? These are the 3 nitrogenous ways, and we know how to form them. We went through that. Are you comfortable with that? You go with that? Okay. Okay. Too much gout. What's the connection between salt and hello kidney? What's the connection there? If I have more salt in my blood, what will happen? More water will be reabsorbed into my blood. What happens to my blood volume? Increases. And if I have more blood to push around, then my blood pressure will go up. Right? Because the more salty my blood is, the more hypertonic it is. Water must flow, right? From the hypo. So it's going to flow into that hypertonic blood. You know, we want water, but if we have too much salt, we can have too much water and our blood volume's too high and our blood pressure's too high. That's why you might be prescribed a diuretic, so you'll pee out some of that water, so you'll make your collecting duct less permeable. All right, so that was that connection. So then let's talk about this, okay? So aldosterone will make your kidneys reabsorb more sodium, which will ultimately make you reabsorb more water. And remember the whole connection that we looked at, okay, is that our afferent arterioles will secrete renin, and renin makes the liver convert angiotensinogen into angiotensin 1. But we're not done, because then who else is involved? Our lungs. They secrete ACE, which makes angiotensin 1 become angiotensin 2. And then you will go to the 2 adrenal glands that sit on top of your 2 kidneys. You hear me on that? Angiotensin 2, right? And you're increasing the tension, maybe you can think of, because you're ultimately going to increase with aldosterone the amount of hydrogen, or sorry, the amount of sodium, which increases the amount of water. And to counteract that, your heart can counteract that. If your blood pressure is too high, then your heart will then counteract that process. And then last but not least, it will help you mature your red blood cells with APBio. Where are you all running to? And vitamin D. Okay, good job. We're all done. Yay, any questions you want me to go over? Okay. All right, have a piece of toast. If it's, you know, if you're really hungry. You know, if you're not hungry, don't have a piece of toast. But if you do, make it sourdough. Yes.
So the thing about earthworms having 2 bladders in the segment, can we use that as a structure in its function?
Yeah, that's a good one. I like that one. Tunephridia, which then it helps them have 2 bladders. Yes. All right, great. You're smart. Remember, your PCR lab isn't due till Thursday, so that's good news. Okay, bye now.